Thank you all for coming. It's really a great feeling to be able to present my work to the leading experts in the field, and it's actually really terrifying also. So I have mixed emotions, so I'm very excited but also very scared. So I am Anika. I am actually a postdoc shared between these two institutions, so University of Zagreb and Genos. I work as a senior research assistant at the University of Zagreb, but I also do my research in Genos. And that research actually revolves around the regulation of IgG glycosylation. So I am going to tell you what I've been doing recently to try to map the pathways that regulate IgG glycosylation. I wanted to give you a quick overview, but I think you know better about this than I do, so I'm going to storm through it really quickly. So IgG is glycosylated on the Fc region, and I wanted to say around 20% of IgG has glycans on the Fab side, but it seems to be a little bit less, so more like 10%. And all of these glycans are, all of these monosaccharide units are added by these main four enzymes, these four glycosyltransferases. And you can see here that the prevalence of all of these different glycoforms that we find on IgG are different. So some are more prevalent than the others. These ratios can actually vary, and we can see that in aging and in different diseases. So different people talked already about this, so I'm going to go through this really quickly. We also know that alternative glycosylation affects the effector function of IgG, so different glycans can then change how IgG interacts with downstream immune pathways. So we know a lot about how glycosylation regulates IgG function, but we are not really sure how the process of glycosylation is regulated itself. So we know it happens in the endoplasmic reticulum and in the Golgi apparatus with the help of all of these different enzymes, so glycosidases, glycosyltransferases, and transporters. But we are not really sure how all of these proteins are regulated. Can they regulate glycosylation of specific proteins? Are there some shared mechanisms? Are there some proteins we haven't identified yet? So there are many very, very interesting questions, and I'm going to tell you about the route we are trying to take to answer some of them and to just gain a bit more understanding about these processes. So our approach is pretty simple. So we combine genome-wide association studies with CRISPR-Cas9 validations. So these are some loci associated with IgG glycosylation, and you can see that there are many, many different prioritized genes. And the idea is to target these genes, to manipulate their expression in cell culture, to see if we can validate that they actually can affect IgG glycome phenotype. And these are the tools that we are using. Is this? Yeah, it's working. Okay, so dCas9 VPR and dCas9 CRAB. So dCas9 is an inactivated version of the Cas9 protein that cannot cut DNA, and it can be fused with different effector domains. Here you can see how VPR looks like. So it is made out of 3 very potent transcription activators that will then activate the transcription of a gene if they are targeted around the transcription start site. And CRAB does the exactly opposite, so it recruits different heterochromatinization complexes that will then inhibit the expression of the gene. And single guide RNAs can be designed to be complementary to any gene of interest, so we designed them to target all of these different prioritized genes. They interact with dCas9, lead them to the sequence of interest, and then our effector domains can do the work. What we have in our lab is this very versatile and very flexible CRISPR-Cas9 toolbox, and it actually enables us to assemble any type of cassette of interest in just one cloning step. So it's very quick and very easy to use. We started with VPR and CRAB, but we now have an array of different epigenetic writers and erasers, so we can now target histone methylation, histone acetylation, even citrullination and phosphorylation, also DNA methylation with TET1 and DNMT3A to induce, again, to either activate or repress gene expression. We can also target many genes at once with these single guide RNAs, so we can design many of them to target many genes to see how will that affect the phenotype of interest. And our phenotype is IgG glycome composition. So to study this, we had to tweak this system a little bit. So we actually combined stable and transient expression of our transgenes. For the stable part, we designed this transposon vector that has these inverted terminal repeats on each side, and also 2 insulators, because we wanted to make sure that our transgene won't be silenced when it's integrated into the genome, so insulators actually prevent heterochromatinization. And then we assembled all of our different modules from the CRISPR-Cas9 toolbox into this transposon vector, and when it is delivered to the cells together with this piggyback transposase, the transposase will bind ITRs, excise the cassette, and insert it into the genome. And when we obtain the stable cell lines that express Cas9-VPR and Cas9-CRAB, the only thing left to do was to deliver guide RNAs, but also to deliver genes for IgG expression, because these are suspension-adapted hex cells and they do not endogenously secrete IgG. And we decided to put them on the same plasmid because then we know that IgG secretion will only happen in cells where targeted manipulation took place. And as a proof of concept, we decided to modulate these main glycosyl transferases. This is the profile we see in our hex cells. So it kind of resembles the one we see on IgG isolated from human plasma, because the most prevalent structures are these ones, the same as we see on IgG from human plasma. And we don't have a lot of sialylation and a lot of bisecting, so we decided to upregulate the expression of MGA3 and ST6-GAL1. We both up and down regulated B4 GALT1, and we down-regulated FUT8 because most of these are core fucosylated. And we were successful in all of these manipulation, meaning that this system does work in this configuration. And when we looked at the IgG glycome phenotype, we could see that changes were actually expected. For an example, B4 GALT1 upregulation resulted in an increase of galactosylation, and so on. And then we were ready to target our GWAS hits. So most of the GWAS hits, GWAS candidate genes, we are not sure how they are involved in IgG glycosylation or in glycosylation in general. And for these 2 ones, the story is a bit more easy to understand because they are both Golgi resident proteins, and that is why we decided to start with them. So GGA2 actually is known to regulate the transport of beta secretase, and we know that beta secretase can cleave ST6-GAL1 from the Golgi membrane. So we did expect to see some changes of IgG cellulation, but unfortunately, after up or down regulation, nothing has changed. For the SBPL3 downregulation, we observed an increase of galactosylation, which was followed by a slight increase in sialylation. And SBPL3 can cleave GTs from the Golgi membrane, so we actually did expect to see this hyperglycosylation phenotype when we downregulate SBPL3. Looking at that profile that we see in free cell cells, the most abundant glycoforms are the ones that are galactosylated, so we decided to focus on IgG galactosylation. And this is one really, really big GWAS studies that was done on IgG galactosylation that revealed over 16 significantly associated loci with 37 prioritized genes. And you can see that B4 GALT1 was the major hit. But of course, there were some other genes for which we have no idea how they are involved in IgG galactosylation. And we prioritized these 7 and targeted them in our system. And this is what we have observed. So when we were targeting this HIV binding protein, in all the experiments we did, we saw that only G2 glycans increased. But we were never able to confirm that we actually upregulated this gene. So this region could be involved in galactosylation somehow, but we don't know if this gene is really the one that does something. When we upregulated TNF receptor family member, we actually saw an increase of G0 glycans. And this protein is actually involved in antibody secretion. So we did hypothesize that maybe it could somehow control the secretion of certain IgG glycophores. When we upregulated EEF1A1, we observed that only G1 glycans go down, and this is an elongation factor. But it can also serve as a signal transducer during inflammation, and it can increase the expression of interleukin-6, and interleukin-6 can decrease IgG galactosylation. So this is something that we also observe in cells. And when we upregulated MAMBA, mannose beta, which is involved in N-glycosylation, only G1 glycans were decreased, and this was repeated several times. Even though we know this gene is involved in glycosylation, we have no idea how it could be affecting only G1. But the interesting thing is that the structure that was actually decreased is that GP9 that Ivan was just talking about. And GP9 is a predictor of cardiovascular risk, and MAMBA is a known risk gene for cardiovascular events. So this is a really interesting association that we want to follow up. But what we knew is that we needed to upgrade our cell culture system to actually perform all of these follow-ups. And the first thing we did is we made this second generation of the CRISPR-Cas9 expression system with the help of this glycan switch grant and the people from LUMC. So we received from them optimized IgG sequences with enhanced nuclear localization signal, so we can improve the production of IgG in our cells. So using our old system, we can only actually reliably quantify those most prevalent IgG glycoforms. And with this system, we got a big increase in IgG production. What we did from our side, we reduced the plasmid size by around 20%, increasing transfection efficiency. And all of these interventions resulted in a 10-fold increase in IgG production. And with this system, we are now able to pretty reliably quantify those sialylated and bisected glycans that are really not that abundant in our cells. Another thing we did to extract a bit more data from the system, we introduced the Fab glycosylation site. So we can simultaneously analyze Fab and Fc glycosylation when we manipulate our GWAS hits. So this is how the glycans look like. So on the Fc, we now have the same profile with this new system, but a lot more IgG, so we can see a lot more different glycoforms. And here is the profile of the cells when we add Fab glycosylation. Unfortunately, there are some really crazy structures because these are hex cells, so there are some antennary fucoses and GalNAcs, which makes the analysis a bit more complicated, but we are working on this, and we can also separate the Fc from the Fab regions to analyze glycome phenotypes from the both regions from the same sample. And with this new system, we were able to validate all of these genes that are in green squares. All of the ones that are in gray squares, we targeted them in our system, but we could not see any change of the IgG glycome phenotype. I don't have the whole data set to talk really about specific changes we see, and this is something that I'm still struggling because I'm not sure what is actually changing, and I'm not sure what is the best way to analyze this data, so it would be great if we could discuss this tomorrow on the meeting. And while we were doing this, a really interesting story popped up that we did in collaboration with Dr. Scholl and Professor Mineo from the Texas University Medical Center. They were looking at the associations between obesity and IgG glycosylation. So prior studies already showed that there is some association between obesity and sialylation. And here, the participants were actually adolescents because they had no confounding things beside obesity that maybe impact the results. And you can see here that, again, like in the prior studies, sialylation was decreased in obese participants and also galactosylation. And when we looked into the data a bit more deeper, we could see that in females, these changes were a lot more pronounced. And to see what was happening, B cells were isolated from a portion of these female participants, and RNA sequencing was performed. And of course, there were many, many differentially expressed genes in obese and lean participants, and they were involved in many different cellular pathways. But what we did, we cross-referenced this data set with our genome-wide association study, the biggest one, and these two genes popped up, and they are actually prioritized for the same region. And this is the region of interest. You can see it here. And then using just qPCR, we wanted to see is the expression of all of these genes changed. And the biggest change was actually observed for VNT3, which was really, really upregulated in obese participants. Then we looked at different glycosylation-related genes to see how their expression was changed. And you can see here that the expression of neuraminidase was increased, and expression of glucosamine epimerase was decreased, which was in line with the observed changes of IgG sialylation. And expression of B4GALT1 and B4GALT2 were also decreased, which is in line with the observed IgG galactosylation changes. And this was followed up in mice. So these were mice that were put on a high-fat diet, and similar things were actually observed. But the most important one is that, again, VNT3 was really, really significantly upregulated in obese mice. And then we came into the play, and then with our system, we targeted VNT3 to upregulate its expression, and we also targeted B4GALT1 just as a control to see what is happening. And this slight upregulation of VNT3 actually induced IgG hypocellulation and hypogalactosylation. So both of the traits we see in mouse and in human participants were changed in our cell system. And then we looked at the expression of genes, genes that are in the pathway of galactosylation, and you can see a slight decrease of B4GALT1 expression. But we have no idea how VNT3 could affect IgG glycosylation. It is involved in many different pathways, and we need to do some follow-up studies. But we shied away from these studies because we knew that our system lacks the right biological context. So in the last 2 years, I was trying to establish a better system that is based on B-cell models, but this is not really an easy task. So we started with freestyle cells because we saw that their IgG glycome resembles the one we observe in humans. They are really easy to manipulate. You can easily deliver transgenes into these cells. They can form single cell clones, and we knew we could do really big experiments because we have over 80 prioritized genes from the GWAS study, and we need a system in which we can do really large experiments. But then I started to screen different cell lines, which is not an easy task to look at all of these variables, and these three actually turned out to be the most important. So efficiency of transgene delivery is very, very low for different B-cell models. So in our lab, we have different transfection protocols established. We have lentiviral technology, and we have electroporation. And with all of these methods, it is really hard to introduce our transgenes into these cells because our CRISPR cassettes are so big. We actually managed to do it in LCLs and Ramos cells, but with very low efficiency. And the downfall of these LCL cell lines was their inability to recover from any type of manipulation. So these cells they had such a low viability after any manipulation, and they wouldn't recover. For the Ramos cells, they can really easily recover from anything, but their downfall is that they secrete such a low quantity of IgG. So we can maybe do experiments in them, but they would be so hard to do for many genes. And then doing this, I actually stumbled upon these ARC77 cells that are also LCLs, and I actually had no hope for them, but it turned out they are really good for all of these variables. So yeah, I was ready to give up when I came across these cells. So the efficiency of transgene delivery for these cells is actually moderate. So even with electroporation, we could introduce our transgenes. And a good thing is that they are really good at recovering. So even though the electroporation of these cells resulted in a very, very poor viability, like 25% maybe, they could easily recover from that. They secrete a lot of IgG. Their glycome really resembles the one we see on IgG isolated from human plasma. Their cell doubling time is okay for these experiments, and I couldn't believe, but they are able to form single cell cultures. And we cannot do maybe the experiments that are this big, like for the freestyle cells, but I think this is going to be fine. Just to show you how the IgG glycome looks like, so these are some new results. We haven't still annotated the peaks, but it really is similar to what we observe in humans. You can see the ARC77 glycans in red and IgG standard in black. They have a little bit more of this bisecting bookmark, but we can see most of the glycoforms. And for the transgene delivery, this was the major obstacle. So just an example how the electroporation of a really small plasmid looks like in these different cell lines. So when you do it in freestyle cells, you can literally see them fluorescent like 2 feet from the microscope. And when I did this for the Ramos cells, almost no cells were electroporated. LCLs were a bit better, but again, they couldn't recover, and ARC77 were pretty good at this. But this GFP plasmid is like 5 times smaller than the CRISPR cassettes we use, so it is really not easy to deliver something like that to the cells. But we decided to try with ARC77 to develop this system that is almost the same as in freestyle cells. We just had to adapt some things. So you can again see here the transposon technology. I decided to use transposons again. We had lentiviral technology also to integrate it, but I worked with transposons for almost 5 years, so I think this technology is really reliable and really easy to use. So we assembled our cassette into this transposon vector, transfected, electroporated our cells with it, established stable polyclonal cell lines. So this is how the cells look like after electroporation. This is how they look like when we have polyclonal stable cells. I don't know if you can see the fluorescent signal. It's much weaker because this is integrated into the genome. And then we tried to establish a single cell cultures, and this photo was taken 3 days ago. So we actually managed to make a monoclonal stable cell line that expresses DCAS9 VPR from a single ARC77 clone. And the idea is to then deliver guide RNAs on a plasmid, this time with antibiotic selection, because we need to select the cells that have our guide RNAs, because our cells do secrete IgG. When we select them, we have to transfer the cells to a serum-free medium so we can isolate IgG for glycan analysis. This is also an accidental discovery that these cells can grow really well in a medium that is optimized for freestyle cells. And that is one of the reasons why we can actually have a lot of IgG from these cells. And what is next? So I hope in the next year I will have the complete GWAS data set validated in our second-generation CRISPR system, maybe not the whole, but most of it, hopefully, and to confirm some of the results in this new B-cell-based CRISPR-DCAS9 system. And when everything is done here, I hope to perform some functional follow-up studies, both on these ARC77 cells, but also maybe on Ramos and LCLs, maybe using some other approach than CRISPR technology. And this is all. I really want to thank my group from the university and from Genos, because this research would never come this far without their help, and also all the partners on the Glycome switch. They really helped, not only with the technology transfer, but also with all the discussions and everything. And that's it.
Thank you very much, Anika, for the very interesting presentation. Questions?
No, it is more a compliment that you identified as ARH77. I think it's a female B lymphoblastic line, right?
So they thought it was a plasma cell tumor cell line, but it turned out these are actually lymphoblastoid cells, yes.
Thank you very much, very interesting. biology gives me headache often.
Yes.
Right? So here is a headache, and maybe Tom and Renee can also step in here. So TNF RSF13B, right?
Yes, yes.
So this one, if you like, read its canonical story, it's cell surface receptor on B cells,
Yeah.
right? And when it meets its ligand, extracellular buff, right, or April, it keeps B cell surviving, right? But here, obviously, there is no April, there is no buff in the middle. These are not B cells. So, like, what could be the mechanism?
I have no idea. I really don't know. So from all of the data I gathered so far, I have no idea for any of these candidate genes how they are working in my cells. But maybe with this new system and with some functional follow-up, we could actually see what some of them do. But right now,
So maybe the ligand is in the serum used to grow the cells. Is it possible?
no. These are serum-free mediums. It's serum-free, yeah.
No,
Yes.
but still there's separate microsome. Because I think the TNF receptor can still signal because probably there's that. I don't know.
So maybe if you really overexpress it, it starts some baseline signaling, right?
It's a ligand setting, yeah, we got ligand factors.
I don't know, it is a cancer.
Yeah.
It doesn't need so much.
Yeah.
Doesn't look so much.
That's the problem with cancer.
Yes, I have microphone. So, do you hypothesize that this number 8 affects the cardiovascular risks in the women wired the GP9? So, is this the line? Because if that is the hypothesis, we could maybe test it, in a cohort where all the information is available, or just by stratifying whether you have this cardiovascular risk association
Okay.
in females.
Yeah, that would be really interesting, yeah.
More questions? I have a question, Anika. So in your last part of your presentation, you showed the B cell transcriptome where you highlighted the Link3. Did you manage to observe also the beta-4-galP or ST6-gal there in the beta cell transcriptome?
Yes, so let me show you here.
Yes, not at the PCR level, but at the B cell transcriptome. So in the first picture.
So you mean if B4GALT1 and ST6GAL1 were also changed? Yes, so ST6GAL1 no, but this neuraminidase and glucosamine epimerase were changed. So maybe this is why this IgG is hyposialylated, and we did observe changes for B4GALT1 and 2. So they were decreased, so again, in line with that hypoglycosylation of IgG. Yeah.
Interesting. So for the sake of time, thank you very much.
Thank you.
So let's come again. The next speaker is Dinesh Alch from I3S.